Okay. Morning. So today we are going to deal with another level of organization. We started with chemicals that form molecules that they form the organelles, and organelles that form the cells. And then when the cells come together, they will form the tissues, a higher level of organization. So the tissues consist of similar types of cells. We cannot say they are exactly in the same shape and size, but they are similar types of cells. They have similar embryonic origin, and we will see how they have similar embryonic origin in a moment, and they perform common functions. The study of tissues under the microscope, this science is called the histology, and histology is the study of normal tissues under the microscope. If it is the study of abnormal diseased tissues, it is called histopathology or pathology, which is the study of diseased tissue. But the normal tissues that we are concerned with in this course are studied by or through histology. These are the four basic types of tissues. You can see them below. There's the epithelial tissue, the connective tissue, muscular tissue, and the nervous tissue. These are the four basic types of tissues. In our lecture today, we are going to deal with the epithelial tissue, and day after tomorrow it will be about the connective tissue, and then later on we are going to study the muscular tissue with the muscular system, and the nervous tissue with the nervous system. So the two lectures that concern the tissues will be dedicated for the epithelium and the connective tissue. Today we are going to study the epithelial tissue, and the epithelium usually it either forms covers or it forms linings. So it covers the body, like the epithelium of the skin, the epidermis of the skin. It lines some organs and tubes, like the gastrointestinal tract, the lining of the gastrointestinal tract, lining of the urinary tract, and other systems in the body, like the reproductive and the respiratory. And also it forms glandular epithelium. The glands, exocrine and endocrine glands, they are formed, the secretory portion of the gland in particular is formed of epithelial tissue. So the epithelium helps to protect, like the skin protecting. It helps to absorb, like the lining of the gastrointestinal tract, secrete, like in the glands, whether endocrine or exocrine, and excrete, like in the epithelium, which is present in the renal, in the urinary system. The other type of tissue is the connective tissue, and it is for protection and support, like in the bone. The bone is a special type of connective tissue, so as the cartilage. It's connective tissue, but special type of connective tissue. It stores energy, like in the fatty tissue, which is present in the subcutaneous layer, and provides immunity, like in the blood. I'm going to deal in more details about the connective tissue day after tomorrow, when we deal with the in much more details lecture about the connective tissue. This is just an introduction about the basic tissues. Muscular tissue is for contraction, and the nervous tissue, these are nerve cells. They detect the changes in the external and internal environment, and after detecting these changes, which means sensory, they respond by generating impulses, motor. So these impulses will either cause contraction of a muscle, or it will result in a gland to secrete, whether an enzyme or a hormone, and so on. So these are so they are called secretomotor. So it's either motor acting on muscles or secretomotor acting on glands, the responses. Now, as I mentioned before in the first slide here, that the tissues consist of similar types of cells with similar embryonic origin. So this is how the embryo is primarily formed. Like we mentioned, that there is a sperm that fertilizes an ovum, a zygote will be formed, the zygote will multiply, and we will have a group of cells that will organize themselves at the beginning as 3 layers. These are the three basic embryonic layers, or germ layers we call them. And these are the ectoderm. This is the ectoderm here, and then the endoderm, and in between them, the intermediate layer, which is sandwiched in between, is called the mesoderm. So we have the three primary germ layers are the ectoderm, mesoderm, and the endoderm. The epithelium, the epithelial tissue, is derived from the three of these layers. Like, for example, the endoderm provides the lining of the gastrointestinal tract. It's derived from the endoderm. The lining of the body cavities, like the pleural cavity, the peritoneal cavity, is derived from the mesoderm. That's why this epithelium is called mesothelium also. And the ectoderm provides the skin, provides the epidermis of the skin. The epithelium of the epidermis of the skin is derived from the ectoderm. Regarding the muscular tissue and connective tissue, it is derived from mesoderm, and the nervous tissue, it is derived from the ectoderm. So it's only the epithelial tissue that is derived from the three layers, depending on the location of that epithelial tissue. So again, we are dealing today with the main basic tissues, mainly the epithelium. And the tissue, by definition, it's a group of similar types of cells that have similar embryonic origin and perform a common function or functions. The cells in the epithelium and in some other tissues, like muscle, for example, but not in connective tissue in general, but in the epithelium in specific, the cells are connected to each other. They are close to each other, and they are connected by junctions. There are different types of these junctions, and so we have 5 types: tight junction, adherence junctions, gap junctions, desmosomes, and hemidesmosomes. Now, the tight junctions, they are strands of transmembrane protein. You can see them here. Remember the cell membrane, which has proteins. Proteins are integral proteins, peripheral proteins, and glycoproteins. The integral proteins, they could be transmembrane protein. They extend the entire thickness of the cell membrane. So these transmembrane proteins of adjacent cells, they will come close together and fuse with each other. And so this will result in a tight junction. There is no gap in between them. It is tight. There is no extracellular space, no interstitial space here. And so they are very tight. They will seal these extracellular spaces. And so we need them in places where we want to avoid leakage of substances. For example, like the cells that line the urinary bladder, they should have tight junction to prevent the leakage of urine outside, or the cells lining the intestine to prevent the leakage of the material to the outside. So these are the tight junctions. They are membrane proteins of 2 adjacent cells tightly fused together. The adherence junctions, again, they are transmembrane proteins, but here the difference is that these proteins, they are connected with each other, and at the same time, from the inner side, from the outer side, they are connected to the protein of another cell membrane, and from the inner side, they are connected to the cytoskeleton. Remember, we mentioned about the organelles of the cell last time. In the cytoskeleton, we have microfilaments, we have intermediate filaments, and microtubules. So these adherence junctions, they are attached to the microfilaments which are present in the cytoplasm from the inside, and so they will retain the shape of the cell and prevent separation in between the cells. They prevent this. When there is tension applied to the cell retains its shape, and it doesn't resist the separation in between the cells. For example, the lining epithelium of the small intestine, the wall of the small intestine, in the wall, apart from the lining epithelium, there are smooth muscle fibers that cause motility. This is what we call peristalsis, in order to mix the contents and to propagate them from one place to another. This peristaltic activity might cause tension on the lining epithelium, and the cells tend to separate from each other, but the presence of these junctions in between them will help in maintaining the cells together. The tight junction will prevent the leakage of the contents into the outside. So each one of these junctions has a special function and a special structure. And they could be present in the same cell. It doesn't mean that each type of cell has only one type of junction. More than one type of junction could be present in the same cell. The other type is gap junctions. As the name indicates, there's a gap in between or a communication in between the cells. Again, they are integral proteins. Remember that the integral proteins, they form the pores in the cell membrane that facilitate diffusion. So these pores, when they come together from two adjacent cell membranes, we will have a gap junction. And therefore, the gap junction allows some substances, small molecules in particular, to pass from one cell to another. And this is important for signaling that takes place in between the cells during embryonic development. These cells, they have gap junctions. Also, ions might pass from one cell to the other, like the gap junctions that are present in the cardiac muscles. We will deal with the muscular tissue later on, and you will see that the cardiac muscle fibers have gap junctions in between them, and this will help in the propagation of the impulse from one cell to another, because the cells can communicate between each other through these small, tiny gaps. The other type of junction, the fourth type, is called the desmosomes. And this is almost has the same structure as the adherens junction. In the adherens junction, there are microfilaments attached to the inside, but here there are intermediate filaments, as you can see here, attached to the inside of the integral protein, and the integral proteins are attached to each other from the outside. I mean, the integral proteins of two adjacent cells are attached to each other from the outside, and again it resists separation. But here we have intermediate filaments. Remember that intermediate filaments are stronger than the microfilaments. It was mentioned last time when we dealt with these organelles. So they are stronger, they can resist more. So they are present in places where there is more tension applied to the cell. The most abundant example of these desmosomes, or the presence of these desmosomes, is in between the epithelial cells of the epidermis. And I will repeat that and mention it again when we deal with the skin next week. So these are the desmosomes. They separate, they resist separation between cells, and they are stronger than the adherens junctions. The other type of junctions are called hemidesmosomes, and as the name indicates, hemi means half, so it's half a desmosome, and it is these hemidesmosomes are present on the basal part of the cell. This is the cell. The epithelial cell has an apical part, or apical membrane, and a basal membrane, and the epithelial cells, as we will see in a moment, they rest or they lie on a membrane which is called the basement membrane, a connective tissue membrane, and they are attached to the connective tissue membrane by a desmosome. They are not attached to the other cell to have the other half of the desmosome, so they will have only half of the desmosome. They will have hemidesmosome, not a desmosome. If there are two cells together, then half with another half will form a complete desmosome. But here it is one epithelial cell with connective tissue, so it has a half desmosome. And this is the only location where these hemidesmosomes are located. The basal layer of the cell, of the epithelial cell, where it lies on the basement membrane. Now these are the general features of epithelial tissue, and we're going to compare that with the connective tissue on Friday when we study the connective tissue. You will find that in the epithelial tissue, the cells are tightly packed together. They are connected to each other, tightly packed to each other, there is a very little interstitial space if you compare that with the connective tissue. So it is very limited interstitial space or extracellular material. The cells, as we mentioned, they lie on a basement membrane, and I'm going to talk about the basement membrane in a moment. So they lie on a basement membrane of connective tissue, and they are avascular, means that they don't have blood vessels. See here in this diagrammatic representation, the blood vessel is present in the underlying connective tissue. So they, while there are no blood vessels in the epithelium itself, the epithelium depends on the spread of nutrients or the passage of nutrients from the connective tissue. Nutrients are brought by blood vessel, and they will diffuse through the connective tissue and the epithelial tissue. But there is no blood vessel in the epithelium or the epithelial tissue itself. However, they have nerve terminals. They have nerve supply. Like the connective tissue, they have nerve supply. So the connective tissue has nerve supply and blood supply, but the epithelial tissue has only nerve supply. It doesn't have a blood supply. And these epithelial cells, they can rapidly divide. We will see a very good example is in the skin. The cells are rapidly dividing, and they can renew themselves, they can replace themselves, they can regenerate, because they have the ability to rapidly divide. Like unlike, for example, some other tissues, like for example the cardiac muscle. When the cardiac muscle dies, it can only be replaced by fibrous tissue. The other cardiac muscles cannot compensate for that. So it's not all the tissues can replicate, but the epithelial tissue has a great capacity to divide and renew itself. The basement membrane that has been mentioned several times until now is formed of two layers, a basal lamina and a reticular lamina. The basal lamina is connective tissue containing mainly collagen fibers, and these are secreted by the epithelium itself. And the reticular lamina contains another type of collagen fibers, tiny collagen fibers called reticular fibers, and it is secreted, or it is produced, by the cells of the connective tissue, mainly the cells that we're going to study next time, called fibroblasts, the cells that produce fibers. Fibroblasts, the cells that produce fibers. So they produce these reticular fibers that are added to the reticular lamina. And as you can see here, that this basement membrane is essential. It anchors the epithelial cells to the connective tissue, attaches them, and the attachment is by hemidesmosomes, as we have just mentioned. And it forms a surface along which cells migrate, especially during embryonic development. So they migrate on this surface, they move on that surface, and they restrict the passage of large molecules. They are restricted. Sometimes in some diseases, this basement membrane becomes thickened, very thick, so it will even restrict the passage of small molecules. Like what happens, for example, in diabetes mellitus, there is thickening of the basement membrane of the capillaries supplying the kidney or supplying the retina, for example, in the eye. And because of the thickening of the basement membrane, then nutrients will not be able to reach these the organs, and so with the patient has as a complication of diabetes, they will have renal failure or blindness. But the main pathology is in the basement membrane. There will be thickening of the basement membrane that will restrict the passage not only of the large molecules in this case, but of the small molecules, reminding you that the epithelium has no blood supply. It requires substances to diffuse from the underlying connective tissue. There is no blood in the epithelium itself. The epithelial tissue can be classified either by the arrangement of cells into layers. So it's either simple or stratified or pseudo-stratified. Simple, as the name indicates, it's a single layer of cells that forms a sheet, a single sheet of cells. Simple epithelium. Stratified epithelium, multiple layers. 2, 3, 4, 5, multiple layers of epithelium. So it is called, this one is called stratified epithelium. The third type of epithelium is called pseudo-stratified. Pseudo means false. So it's actually, it is not stratified, but it looks as if it is stratified. The reason for that, as you can see here, that all the cells, they lie on the same basement membrane. But some of the cells are longer than the other. They are taller than the other. So not all the cells reach the apical surface. Some of the cells do not reach the apical surface, but they all stand on the same floor. But some of them are tall, some of them are short. And so the nuclei, which we usually, when we look at the tissue under the microscope, the dark structure, the nucleus, attracts our attention. So the nuclei, because the cells, some of them are tall, some of them are short, the nuclei are present at different levels. So we think that it is stratified, but actually it is not stratified. That's why it's called pseudo-stratified epithelium. All the cells rest on the basement membrane, but the nuclei are found at different levels. So it gives the impression as if it is multi-layered. So simple, stratified, or pseudo-stratified. And the other classification is the shape of surface cells. So depending on the shape, it's either squamous or flat, like the tiles, or it is cuboidal, or it is columnar, tall columnar or rectangular cells, or it is transitional. In transitional, it means that the surface cells, they change their shape. And this is a characteristic epithelium that is present in the urinary system. So that's why it's called urothelium. Like in the lining epithelium of the urinary bladder, the apical cells, the apical cells, usually they are cuboidal, but if the urinary bladder is empty. But if the urinary bladder is distended, then this will cause tension and results in flattening of the cells. So instead of being cuboidal, they will become flattened. So that's why the epithelium is called transitional epithelium. And as I mentioned, it is the characteristic epithelium of the urinary system. Yes?
Where in the body would you find pseudostratified cells? Or
Yeah.
would they be anywhere?
Yeah, I will show you that in a moment. Pseudostratified epithelium is a characteristic epithelium of the respiratory system, especially the upper respiratory system. And that's why sometimes they call it respiratory epithelium. Okay? Yeah. So, a second classification, as I mentioned here, it's according to the shape of the cells, but which cells? It is the surface cells, not all the cells, the surface cells, in case of a stratified epithelium. Now, the simple squamous epithelium, it is simple, means one layer of flattened cells. They have nuclei, as you can see them here. They are flattened oval nuclei. And if you look at the surface of the epithelium, it looks like tiles. From the side, they are spindle. They look like spindles here. And again, the nucleus is flattened, the oval and flattened. This is very thin. And being very thin epithelium, because it is simple, so it's one layer, being very thin, it means that it cannot be present on external surfaces where there is much tension is required. And being thin, it means that it allows easy passage of substances through the epithelium. So that's why this epithelium controls filtration. For example, it is present in the kidney to control the filtration of the blood into the tubules of the kidney. It also allows diffusion of gases, so it's present in the lining of the alveoli of the lung because it's very thin. And as I mentioned, being thin means that it cannot be present in the surfaces which are on which tension is applied. They line blood vessels, and they are called endothelium, and they line body cavities, as I mentioned before, like peritoneum or pleura, and they are called, in this case, it's called mesothelium. It's called mesothelium because this type of epithelium is derived from the mesoderm, while the endothelium that lines the blood vessels is derived from endoderm. Remember that the epithelium can be derived from mesoderm, ectoderm, and endoderm, unlike other types of basic tissues. And also it is used for secretion, like in the serous membranes, the peritoneum. You will study the peritoneum later on, and the peritoneum is a membranous tissue, serous membrane. There are two layers, and there is a fluid, serous fluid in between them to reduce friction. And this fluid is secreted by the lining epithelium, so they secrete as well. This lining, simple squamous epithelium, secretes to reduce friction in between the two layers, because the intestine is always moving. So in order to facilitate this movement and reduce friction, there is this type of a peritoneum that is lined by simple squamous epithelium. The other type of epithelium is the simple cuboidal epithelium. Simple, again, I repeat, means one layer of cells. Cuboidal, the shape of the cell is cuboidal, and here, because the cell is cuboidal, it is not flattened, therefore the nucleus is rounded and it is centrally located. Unlike here, the nucleus is flattened and become oval because it becomes flattened. But when the epithelium is taller, then the cell is taller, then the nucleus become rounded and central in the cell. And these are located in the tubules of the kidney, and the most important example here is that they are present in some of the glands, like the thyroid gland. They are the secretory cells of the thyroid gland. And so they function in absorption, like in the kidney tubules, and in the secretion of hormones, like in the thyroid gland. The other type is simple columnar epithelium. Again, simple means one layer. Columnar, the cells are elongated, rectangular, tall cells, and it could be either ciliated or non-ciliated. I'm dealing now with the non-ciliated. So it is a single layer of elongated cells, and the nucleus is oval, and usually it is toward the base, toward the basal surface, not in the center, but toward the basal surface. And some of these cells, they might have microvilli. Remember, the microvilli was mentioned last time. They are multiple finger-like projections of the cell membrane, and they are present on the apical surface of the cell to increase the surface area. For example, this is here in the intestine, the small intestine. They increase the surface area for absorption. So they might have microvilli. And some of these cells in between are called goblet cells. Again, they are tall columnar cells, but they secrete mucus. And this mucus collects in the apical part of the cell. Remember, the nucleus is in the basal part of the cell. The mucus collects in the apical part of the cell, and it is going to be secreted later on. But what happens when we study the tissue under the microscope is that during tissue preparation, this mucus, which was collected in most of the apical part of the cell, will be washed away during tissue preparation. So we look at the cell looks as if it is empty, and the shape of that where the mucus was located looks like a goblet. So that's why these cells are called goblet cells. It is another type of non-ciliated, simple columnar epithelium present in the gastrointestinal tract, for example, the stomach and the rectum. They absorb, and in addition to that, they secrete mucus by the goblet cells, because this mucus will protect and reduce the friction, especially in the large intestine, where the contents of the large intestine become harder and more formed. So we need more mucus in order to reduce the friction. So they absorb and they secrete. Simple columnar non-ciliated epithelium. The other type is the ciliated, simple columnar epithelium. They have cilia, and you have to differentiate between microvilli and cilia. Microvilli are finger-like projections that are non-motile. Cilia are motile. motile projections from the cell. They are short, multiple motile projection. Motile because they contain the microtubules, another part of the cytoskeleton of the cell. So they contain a bunch of microtubules, and they become motile. Again, they will have an oval nucleus that is located toward the base of the cell. They might have goblet cells, like the ones that are present in the respiratory passages. So let's say, for example, in the respiratory passages, in the smaller, in the bronchioles, narrower, not the higher respiratory passages, but in the small respiratory passages, this simple columnar ciliated epithelium has goblet cells that secrete mucus. The mucus will form a surface layer, and the cilia are motile. They have a sweeping action. Particles, like for example in the smoke, the particles will be trapped in the mucus, and then the mucus with the trapped particle is swept away. Okay, and so for example in a smoker, the mucus will become thick, and the cilia will be destroyed. That's why smokers cough, because they want to remove the mucus. The cilia are not working well. The same thing happens in the fallopian tube, in the uterine tube. In the uterine tube, the lining epithelium again has cilia, and these will move the fertilized ovum. The ovum is fertilized in the tube, and then it will be implanted in the uterus. So the presence of the cilia will move the fertilized ovum as it is multiplying and move it to the uterus. If these cilia are destroyed, like for example infection destroys the lining epithelium and destroys the cilia, then the fertilized ovum stays in the tube, and this results in what we call ectopic pregnancy outside the uterus, which is in this case pregnancy in the tube, and has another name. It's called the tubal pregnancy, because these cilia are not functioning well. The pseudostratified epithelium, columnar epithelium, is as I mentioned, multiple levels of nuclei, but single layer of cells. All the cells, they rest on the same basal lamina, and may or may not have cilia, may or may not have goblet cells. The respiratory epithelium has cilia and it has goblet cells. The upper respiratory passages, like the nose, the trachea, the bronchi, all of them are lined by pseudostratified columnar ciliated epithelium with goblet cells. Yet there is pseudostratified columnar epithelium with no cilia, no goblet cells, and this is the kind of epithelium that is present in the epididymis. We are going to study the epididymis later on. Epididymis is a long, convoluted tube that is located close to the testis, and it helps to propagate the sperms from the testis to the vas, but also it acts as a storage place for the sperms. So here it is lined by pseudostratified columnar epithelium, non-ciliated epithelium. This epithelium absorbs to concentrate the sperms. Now the stratified epithelium, we have stratified squamous epithelium, and the best example here is in the skin as a stratified squamous epithelium. However, this stratified squamous epithelium could be in two types. Either it is keratinized or non-keratinized epithelium. It is called stratified squamous epithelium because the surface cells, the apical cells, are squamous. But if you look at the basal cells, they are cuboidal or even columnar cells. But we call the epithelium, the stratified epithelium, according to the shape of the apical cells. Here, again, it has the same features of epithelium. The cells multiply and replace themselves. So the cells in the basal layer, they multiply and they replace the surface layer of cells. The cells produce, when they reach the surface layer, they die. The keratinized type of epithelium means that the surface layer, they are dead and contain keratin, which is a type of tough protein that is used for protection, or it could be non-keratinized stratified squamous epithelium. The non-keratinized stratified squamous epithelium is present in the beginning of the GI and the end of the GI. Like it's present in the mouth, and it's present in the lower part of the anal canal. It is present in the vagina. It is present in the outer surface of the cervix of the uterus, for example. So in there it is used for protection. And also the keratinized, the very obvious example is the skin, and the function of the stratified squamous epithelium is obviously protection from against abrasion, water loss, microorganisms, and protection against harmful ultraviolet radiation like in the skin. So when there is any wound in the skin, then or abrasion in the epithelium of the skin, then the skin is more vulnerable for infection. If there is burn, for example, there is loss of water from the body resulting into dehydration because it prevents water loss. So the most common type of stratified epithelium is the stratified squamous epithelium, whether keratinized or non-keratinized. Very rare types of stratified epithelia. We have stratified cuboidal, stratified columnar. They are rare, and usually they are present in the ducts of glands, large ducts of glands. The other type is the transitional epithelium, and I think that by now you know that the apical cells of that epithelium, they can change their shape according to the tension of the organ that they line. An obvious example is in the urinary bladder. That when it is full with urine, it becomes tense, the tissue is stretched, and the epithelium changes, the surface epithelium changes from cuboidal into more flattened. As I mentioned before, that the epithelium either covers or lines, but also it is present in the glands. So the glandular epithelium, we have two types of glands. We have exocrine glands and endocrine glands. The exocrine glands, as the name indicates, they secrete to the outside, exo. So they secrete to the outside, and so they have limited effects because they have ducts and they secrete, and the effects of the secretion is local. But for example, like sweaty glands, like salivary glands, these are exocrine glands. So they have local effects. They have ducts and secrete to the outside. The endocrine glands, on the other hand, they secrete, their secretion goes to the capillaries, goes to the blood, so it circulates. So it has far-reaching effects. Like for example, we have the pituitary gland, which is present in the brain, lower part of the brain. It secretes into the blood, and the hormones that it secretes might affect the thyroid gland, might affect the testis, the ovary, might affect the suprarenal gland, far away. But the sweat glands, when they secrete, they will cause, they will deposit the sweat on the surface of the body locally only. They cannot affect other structures. And there are mixed glands. An obvious example of a mixed gland is the pancreas. The pancreas has part of it is exocrine and part of it is endocrine. The endocrine part of the pancreas secretes hormones that control blood sugar, like insulin and glucagon. And these are secreted into the blood. But the exocrine parts of the pancreas, it secretes digestive enzymes. And these digestive enzymes will go through a pancreatic duct that opens into the small intestine. And so these enzymes, digestive enzymes, will produce their effects on the contents of the small intestine. Local effects, not far-reaching effects like the hormones that are secreted by the endocrine part of the pancreas. So exocrine, endocrine, and mixed glands. The glandular tissue is epithelial tissue. That's why we are studying it here. Glands are also classified according to their shape and structure. So they are either unicellular, like the goblet cells, one cell secretes, and so it's a gland, or multicellular. So we have most of the exocrine glands are multicellular. They can be classified according to the shape of the duct into simple, where they have one duct, for example here, simple, or they are branched here. So they are simple or branched, The other classification depends on the shape of the secretory portion of the gland. Again, I repeat, the exocrine gland has a secretory part and a duct. The function of the duct is just to transport the secretion from the secretory portion to the exterior. It doesn't secrete. But this is the secretory portion. It could be either like a tube, tubular. It could be like what we call a sinus or alveolus. So a sinus, a sinus. Or it could be partly tubular, partly acinar. So the last slide is classification of exocrine glands according to function. Most of the exocrine glands are called merocrine glands or ecrine glands. The cells will release their products by exocytosis. Remember, we mentioned about exocytosis, that there are vesicles produced in the transport vesicles and the endoplasmic reticulum. They move to the Golgi apparatus. They will be processed and packaged, and then they will form secretory vesicles from the Golgi complex that fuse with the cell membrane, and the product will be secreted. This is most of the glands, exocrine glands in the body, they secrete by this type of secretion, and it's called merocrine secretion. The second type of secretion is apocrine secretion. And as the name indicates, apo related to the apex, the secretion of the gland will be collected, the secretion of the cell will be collected in the apical part of the cell. And then this apical part of the cell will be pinched off, will be detached. So part of the cell will be detached. It's not like a vesicle opening to the outside. The apical part of the cell, apocrine secretion, will be detached. And because these cells are epithelial cells, they can repair and renew themselves. So there is no problem in that. They can just repair and renew themselves. This is what happens in the mammary gland. Secretion of milk, especially the fatty component of the milk, it is secreted by apocrine type of secretion. Most of the secretion of the exocrine gland is merocrine, and there is another type of secretion, holocrine. In holocrine secretion, the secretion of the cell will be accumulated inside the cell, and then the entire cell will explode, will die, and release its secretion. And this is what happens in the sebaceous glands of the skin. So it's called holocrine secretion. The whole cell explodes, and the secretion, after being accumulated inside the cell, it will be released to the outside. So we have merocrine, which is the most common, apocrine, the apical part pinched off, or holocrine secretion. Any questions?
I have a question.
Yes?
Is the base membrane the same as the base membrane, or are they different?
It is the same.
It's the same. Okay.